Hey guys, your video today is going to be a little bit different than usual because instead of just giving you some notes, I'm actually going to tell you a little bit of a story. So welcome to Story Time with Mrs. Koester. And the name of the book that we're going to be reading today is Not Your Grandma's Protein Synthesis. So today I'm going to bounce back and forth between telling you a little bit of a story and then talking about how it relates to what actually happens in protein synthesis. There is a notes organizer for you to fill out as you're watching this video, so please make sure you are doing that. There's a diagram on the last page that I'm going to put on the screen, and you need to make sure that you label exactly what I have on the screen on your piece of paper, because this is definitely stuff that you're going to be expected to know. Now, protein synthesis is one of the more complicated topics that we cover in freshman biology, so hopefully this little story time will help you gain a better understanding of what's happening in this complicated process. So again, this is the diagram you're going to see on the last page. So anytime you see this diagram pop up, make sure that you are filling it in on your sheet of paper. So before I get into the story time, let's just review a couple of things that we should know about proteins. You should know from our biochemistry unit that proteins are a biological macromolecule that are made up of chains of amino acids. You memorize that during the biochemistry unit. So now, as part of this unit, you're going to understand really what that means and how it happens. Why do we care about proteins? Why do we care about how they are made? Because we know that proteins are diverse and abundant throughout all living organisms and that they have a wide variety of functions. They're involved in metabolism, speeding up reactions. Remember, those are enzymes, which are proteins. They're used for building muscle. They're used in DNA replication. They're used in cell transport, and on and on and on. Lots of different functions. Now, hopefully you know by now that the instructions for the building of a protein are carried on what we call a gene. And so a gene is a specific portion of DNA which contains the instructions for the making of a protein. So I want you to think of DNA as like having the recipe for the building of a specific type of protein. Okay, so let's go ahead and label two things on our diagram. First of all, the DNA and where that molecule is found, and hopefully we know that in eukaryotic organisms, DNA is found in the nucleus. So instead of labeling the nucleus, we're going to actually label the outside of the nucleus, which is called the nuclear membrane, because that's going to be important in our protein synthesis process here. Okay, so on your diagram, label DNA and label the nuclear membrane. Okay. So now let's start our story. And the reason it's called Not Your Grandma's Protein Synthesis, because the main character of our story is in fact my grandma. This is a picture of my grandma, and she is what I call the ultimate grandma. Like when you think of a grandma, you're thinking of my little grandma. Look at her, she's adorable. She's got her white little hair, and she's just what you picture when you think of a grandma. She loves having her family over and cooking for them and feeding them and providing for them, and she loves nothing more than spending time with her family and her grandkids. Now, one of the things that is special about my grandma is that she has a ton of recipes that our family loves, right? Recipes that have been passed down from her mother and her grandmother and then have been given to our family, and we consider them very special recipes in our family. But we really live for her chocolate chip cookie recipe. This recipe, guys, is award-winning. Best chocolate chip cookies you've ever had. Like crunchy on the outside, hot and warm and melty on the inside. Delicious chocolate chip cookies. So I want that recipe for the chocolate chip cookies because I love making my grandma's chocolate chip cookies. But there's a problem. My grandma, the ultimate grandma, lives in Oklahoma, and she's getting kind of old and she doesn't really like to travel much anymore. Airports are like a little too stressful for her. So the problem is that my grandma doesn't travel out of Oklahoma anymore. Yet I am here in Georgia and want to make the chocolate chip cookies. But my grandma, she would never part with her precious original handwritten recipes that have been passed down to her, right? She's not going to let those go. But what she did do was many years ago, she allowed my mom to copy down the chocolate chip recipe from her original notebook onto, a little index card. Why are recipes always on index cards? So years ago, my mom copied down the recipe onto an index card and brought that recipe and gave it to me in Georgia. Okay, so we're going to stop here in our story and we're going to talk about how does this little story that I'm telling you, how does this sort of like what's happening in protein synthesis? So if you remember, proteins are made in the ribosomes of cells, but we just talked about how the instructions for the building of a protein are found on DNA, and DNA is found in the nucleus. So the instructions for the building of a protein are found in DNA. The problem is that DNA is too big to leave the nucleus. So the instructions for the building of a protein are stuck in the nucleus because they are physically too big to move out of the nucleus. So how do we solve that problem? We copy down the recipe, just like my mom did years ago. The way we copy down the DNA recipe is by using a molecule of messenger RNA. And if you remember, RNA is single-stranded. It is literally physically smaller than DNA. And because it is single-stranded, it can actually leave through the nuclear pores of the nuclear membrane. So messenger RNA basically copies down the DNA recipe and carries that recipe for the building of a protein from the DNA in the nucleus to the ribosome where the protein is actually going to get constructed. That process of copying down the DNA onto a molecule of messenger RNA is called transcription. Okay, so go ahead and make sure you have these notes here. The purpose of transcription is to copy the DNA message onto a molecule of mRNA. Why do we do that? So that the instructions, the recipe, can leave the nucleus and make its way to the ribosome, which is where it's going to be turned into a protein. So how does this happen? It happens with enzymes, just like we learned about in replication. DNA helicase has to unzip the gene, has to expose it, and RNA polymerase is going to construct the molecule of messenger RNA. The messenger RNA is going to leave the nucleus, and it's going to travel to the ribosome where it's going to get what we call translated, and we'll talk about what that means in just a second. So now in our story, it's time to start following the recipe and making the actual food. That's translation, the building of your protein. But let's go ahead and label on our diagram the process of transcription and the molecule that's being created in transcription and label it on your diagram. Okay, so the process taking place, you should have transcription. The molecule being created is what we call mRNA or messenger RNA. So go ahead and write that down as well. All right, so back to our story. Let's just quickly review who our key players are in our story and what they represent in real life. So my grandma's recipe book that has all of the recipes, that would represent a molecule of DNA with lots of genes on it, right? So the whole molecule of DNA is like her big giant notebook full of recipes. Each individual recipe, or in this case, my grandma's recipe for chocolate chip cookies, represents a specific gene. So a gene carries the recipe for the building of one protein. Oklahoma kind of represents the nucleus because my grandma's recipes are kind of stuck there. My mom represented the messenger RNA, right? She copied down the recipe and brought it to Georgia where I am going to make the chocolate chip cookies. So she kind of represented the mRNA. And then my mom doing the physical act of copying that recipe, that sort of represents transcription. Okay, so what happens next in our story? Here's the thing about me, I love to bake. I don't really like cooking, but I do love baking. But to me, baking is not very fun alone. I love to do it with my children, but I really, really like to do it with my sister, especially around the holidays. We love to bake together. So meet my sister. Now, here's the thing. I am the older sister. My sister is the baby of the family. So the beauty of being a big sister is that you kind of get to boss your little sister around. Even when you're grown adults, it still happens. So here's what baking with my sister looks like. We're in the kitchen together, and I'm the one standing at the bowl reading the recipe. And so I read the recipe one line at a time. I'm like, okay, I need 3 eggs. And I call out those instructions to my sister. I need 3 eggs. And so she runs to the refrigerator, gets the eggs, and brings them back to me. And then I'm like, I need a cup of flour. So she goes to the pantry and she gets the flour and then she brings it back to me. She's really the one doing all the work, right? Running all around, getting all the ingredients. I'm just the one assembling the ingredients and reading the recipe. That's my job. So how is that like what's happening in real life? What I just sort of mentioned in my story is very similar to what's happening in what we call translation. So translation is where we are reading that message that was on the molecule of messenger RNA and turning it into our final product, which is a protein. So the mRNA message is read by the ribosome 3 nitrogen bases at a time. We call that a codon. So one codon is 3 nitrogen bases, and one codon signals for a specific amino acid. So just like the line on the recipe says, I need 4 eggs, That's like one line is one codon. And then the amino acids are like my eggs, right? My ingredient that needs to get put together and assembled to make a protein. So let's go ahead and fill out a couple of things on our diagram here. We're gonna remind ourselves that 3 mRNA bases make up a codon and that each codon signals for a specific amino acid. So over here on your diagram, go ahead and fill in codon and amino acid. And then we're going to label an actual codon on the mRNA strand, and we're going to label the amino acid that is being brought to the ribosome, that's being signaled for. Okay, so here's my codon because this is my messenger RNA. So there's 3 bases on my messenger RNA. That is a codon, and that is signaling for this specific amino acid right here. So let's talk about amino acids for just a second. If you remember, there are 20 amino acids, and your body can make somewhere around 10 or 11, depending on how old you are. The other amino acids, which we call essential amino acids, you have to get from your food. Now, here's the thing. There's only 20 amino acids, but there are 64 possible combinations of codons, right? Sequences of 3. So what does that mean? It means that several codons signal for the same amino acid. So you can see on this chart here, TTT or TTC all signal for this same amino acid. So here's one that signals for 4 different codons that signal for one amino acid. So that's why you have 64 possible combinations, but only 20 amino acids, because some of them signal for the same ones. Okay, continuing on about translation. That process of reading the mRNA sequence, 3 bases or 1 codon at a time, and assembling the protein together, that is all called translation, everything that's happening at the ribosome. This is the following of the recipe, reading it a line at a time, and then assembling the ingredients to make the chocolate chip cookies, or in this case, the protein. Where in the cell is this taking place? This is all taking place at the ribosome. The ribosome is reading 3 bases or 1 codon at a time in order to assemble the proteins. So let's go ahead and label translation and ribosome on our diagram here. Okay, so the process taking place here at the ribosome is translation, and then go ahead and label that structure there as the ribosome. This is the thing responsible for reading the codons and assembling the ingredients to make the protein here. So remember, my sister was the one doing all the work. She was the one actually going and grabbing all the ingredients and just bringing it to me. I was really just the one putting it all together where the action was happening, right? But she was the one going out and grabbing whatever was being signaled for, whatever I was calling for. She sort of represents what we call transfer RNA. Transfer RNA is a specific type of RNA that brings or transfers the amino acid that is being signaled for to the ribosome. How do we know it's bringing the right amino acid? Because the mRNA has the codon that signals for that amino acid, and the transfer RNA carrying it has the complementary anti-codon. And they quickly sort of match up with each other, the amino acid gets dropped off, and then the transfer RNA goes away to grab the next ingredient. So you know it's bringing the right one because it has the complementary anti-codon. So let's go ahead on our diagram and label transfer RNA and anti-codon found on the transfer RNA molecule. Okay, so which molecule is seen here? I can always remember which one is the transfer RNA because it kind of looks like it's a T, right? Transfer RNA, tRNA. And there's my sequence of 3 bases that kind of hang at the bottom of our tRNA molecule. So that is my anti-codon. Here's my codon on the mRNA. Here's my anti-codon on the tRNA. Okay, so the transfer RNA or tRNA molecule continues to transfer the signal ingredients, the amino acids, and basically drop them off at the ribosome, kind of like what my sister does at the bowl, right, with me. The amino acids join together with what we call peptide bonds. Think of the peptide bonds as, mixing all the ingredients together. They're holding everything together, forming what we call a growing polypeptide chain. That word should ring a bell to you from our macromolecule unit. So a polypeptide chain is just another fancy name for a protein because it's many peptide bonds holding all those amino acids together. So let's go ahead and label the growing protein on our diagram. This chain represents the growing protein, kind of the whole point of this picture here, right? This is going to continue to happen until we reach what is called a stop codon. There are certain codons that when the ribosome reads it, it means, hey, we're done, right? The protein is done being made. It gets released into the cell wherever it is needed to go. So back to our story, let's just refresh our memory on what some of these key players represent. So I'm standing at the bowl, I'm calling out the recipe one line at a time. So I represent the ribosome, reading one codon at a time of our mRNA message. Each line of the recipe kind of represents a single codon, right? Each line of the recipe calls for one ingredient. So that's sort of like an amino acid. So each ingredient is a single amino acid. My sister, who was the one running around and grabbing the ingredients that were being called for, represents the transfer RNA. And then the ingredients actually, getting brought to the bowl and getting put together, that represents the process of translation. And then the final product, which are my chocolate chip cookies, that represents the protein, the final product, all the ingredients, all of the amino acids put together. So let's go ahead and practice and make sure we understand what this actually looks like in real life. So on your paper here, you have a DNA sequence, GTC, AAT, blah, blah, blah. I want you to tell me what the complementary mRNA sequence would be. So imagine that this was a strand of DNA that has been exposed for transcription. So if you were RNA polymerase, what would be the mRNA bases that you would construct if this was the exposed DNA sequence? I've done the first one for you, CAG. Pause on this and see if you can figure out the rest. All right, so you should have come up with something that looks like this, right? Transcription would transcribe this sequence here onto a molecule of messenger RNA. That would leave the nucleus and go to the ribosome, this strand here, where it's going to get read by the ribosome 3 bases at a time. So now I want you to translate that transcribed message. So if the first codon that was read by the ribosome was CAG, I want you to look on the amino acid chart that you have provided as a reference, either in front of you or on CTLS, and I want you to look up what would CAG signal for. You should get the abbreviation for this amino acid right here. Okay, now I want you to pause on this slide and use your amino acid chart to see if you can figure out what would be the remainder of the amino acids that are signaled for in this particular process of translation. And you should have gotten something that looked like this. Notice that UGA, the greatest codon in the world, is a stop codon. It signals for the completion of the building of a protein. All right, now here's kind of a tricky question for you. If that is your sequence of mRNA codons, what would be the anticodons that you would find on the transfer RNA molecules that are bringing those amino acids that are being signaled to the ribosome? Remember, they're going to complement the codon. So if the codon is CAG, then the anticodon is GUC. Pause on this slide and see if you can figure out the rest of our complementary anticodons that would be found on our molecules of tRNA, bringing in the amino acids during translation. And you should have ended up with something that looks like this. Here's a little cheat sheet hint for you. The tRNA anticodon sequence, if you look back up at our original DNA sequence, will always be exactly the same, except everywhere there's a T, you're going to replace it with a U because we're looking at a molecule of mRNA, right? It's a complement of a complement, so it's going to go back to the original sequence. All right, now, some critical thinking here. What if a mutation occurs? What if instead of our original sequence, we have an A here instead of, if you look back at the original, what's supposed to be a C? This is supposed to be CCT. So what happens if instead the DNA, let's say during replication, builds a strand that has an A instead of that C? Is it going to have an impact? So go ahead and transcribe your mutated sequence here. And you should have gotten this right here. And now let's use our mRNA chart to our amino acid chart to figure out how is this going to change the amino acid sequence that it signals for. And what you should see is that you end up with a stop codon right here at the beginning. This particular mutation, which is called a substitution, inserts a stop codon too early. Is that going to be a problem? Yes, that's going to be a problem because the protein that is supposed to get built never gets built, which is always going to be a problem. All right. Hopefully our little story time today helped protein synthesis, a complicated process, make a little bit more sense to you. That is all I have for you today. Make sure you have all of those notes filled out and that you submit them when you are done. Y'all have a good day.